All right, guys, so what we're going to do today is we're going to talk about erythropoiesis and the lifespan of a red blood cell or an erythrocyte. Well, first off, before we go into that, what is erythropoiesis? Erythropoiesis is the normal production of red blood cells, right? So it's the production of red blood cells. Where does it occur? Because we need to know what it is, where it occurs, and what drives this process. So again, erythropoiesis is the production of red blood cells, and guess where it's happening? It's happening in the bone marrow, specifically the red bone marrow, which is usually found in the epiphyses of long bones, right? So you can find the trabeculae, the spongy bone. That's where you're going to find the actual red bone marrow. And again, that's where erythropoiesis occurs. So it's a production of red blood cells. It's occurring in the red bone marrow. And what drives it? Now, erythropoiesis is usually driven when you want to make more red blood cells. It could be due to blood loss. So maybe you've had some type of stab wound, maybe you have an ulcer, you're losing blood. Another reason could be due to a less availability of oxygen. Let's say that you're climbing Mount Everest and the oxygen levels are low up there. So the low oxygen levels is actually going to trigger your body to want to produce more red blood cells so you have more oxygen. And there also could be other causes such as anemia, and we could go on and on. But the overall cause of this erythropoiesis is usually referred to as hypoxia. Okay? And it's due to low oxygen delivery to the tissue, so an inadequate oxygen delivery. All right, so now, what do we need for erythropoiesis to occur? We need a certain amount of minerals and nutrients and substances for this to occur. So let's go over here to the GI tract and let's go through all the things that we're going to need for this process to occur. So come over here, we got the stomach, right? So the stomach is actually going to be what's allowing for a lot of the substance to come in. Then we're going to have the duodenum right here, and then we're going to come all the way down here to the ileum. We're going to look at the absorption of the first substance that we absolutely need for this process, which is iron. So we have iron right here. Now you can get iron from the diet, right? You can get it from certain supplements. It can come from various different sources. Now, how does this iron relate? We'll talk about that after we show the entire absorptive mechanism. So let's say we bring iron in, right? We bring iron in. When iron is actually moving down through the stomach, it gets over here to about the duodenum. In the duodenum, there's going to be these enterocytes right here. So we took one enterocyte and blew it up. So this is a duodenal cell, or an enterocyte, right? Or an intestinal cell. And it has a whole bunch of different proteins on here. The first protein that we're going to look at here, if I follow this iron all the way down here, it's going to come out here, right? There's a specific enzyme here that basically converts iron from 3+ into 2+. So it went from iron 3+ into iron 2+. This enzyme is a reductase enzyme. We give it a specific name. It's called duodenal cytochrome B. So this enzyme is called duodenal cytochrome B, and it's a ferroreductase enzyme. And basically what it does is it converts iron from the 3+ into the 2+ state. Then there's another transporter. And what happens is this transporter brings the iron into the cell, into this intestinal cell. But it needs H+ in order for this to happen. That's not that bad, right? So look, H+ and iron is actually coming into the cell. And it's moving in, and we need this transporter for this to occur. What is this transporter called? This transporter is called divalent metal transporter type 1. Okay? So what is this transporter called? This transporter is called divalent metal transporter 1. So iron gets reduced into Fe2+, and then it gets taken in through DMT1, or the divalent metal transporter. Then once it's taken in, look what's going to happen here. So we're going to have this iron. We have to bind this iron onto something inside of the cell, because iron, whenever it's actually moving and free-floating within the blood or within our tissue cells, it can lead to free radical accumulation, and that can cause a lot of damage. So we want to bind this puppy to something that's going to prevent that. So look here, there's actually going to be this protein here. And this protein is actually called apoferritin. But what happens is, apoferritin is going to bind onto this iron. So look what happens. This iron comes in, and it actually binds with this apoferritin. And if it binds with the apoferritin, it actually is going to convert into ferritin. So now, look what we have here. We had apoferritin, which didn't have the iron. Now we have ferritin, which is actually binding the iron. Then what can happen? You know these ferritin molecules, they love to combine with one another sometimes. So look at this. I'm actually going to combine a whole bunch of ferritin molecules here together. And again, what does ferritin have within the center? It has iron, 2+, still in the 2+ state. And look at it, it's bound all right there. So again, what is this molecule here called? This molecule is called ferritin. And then what is this polymer of a whole bunch of ferritin molecules? This polymer is called hemosiderin. So look, we have ferritin here, and a whole bunch of ferritin molecules combine together to form hemosiderin. Now, some of this iron, it might not even bind on to the ferritin. Well, it actually can bind onto the ferritin, I'm sorry, but some of that ferritin can actually come over here, right, and drop that iron off to get it out of the cell, because we need it in the bone marrow. So, if it comes and drops this iron off and it lets it out, so now the iron's actually coming out here, right? It goes out through this very, very important transporter. It's called ferroportin, because it's ferroimportant, right? So we need this ferroportin molecule to transport the iron, which is again in what state? It's in the 2+ state. And then what happens is there's another enzyme which is called There's another enzyme here called hephastin. Hephastin. And what does hephastin do? It takes the iron from the 2+ and converts it into the 3+ state. That's so simple, right? So look, what happened? Iron is reduced. Then it's transported in. Then it's bound to ferritin. And then ferritin can combine to form hemosiderin. But some of that iron we need to get into the bloodstream. So what do? We bring it out here through this ferroportin, who is ferroimportant. And then Fe2+ gets converted back into Fe3+. And then we put it onto this transporter into the bloodstream. And again, why must iron be bound to a transporter protein? Why can't it just freely circulate? Because it can undergo what's called Fenton reactions, which is producing tons of free radicals. We don't want that. It's going to cause a lot of oxidative stress. But now it binds onto this protein here. What is this protein called? This protein is called transferrin. Oh, so simple, right? So again, we had the transferrin now, which is bound to the iron. Now what's going to happen to this iron now? We're going to take it all the way to the bone marrow. Or guess where else we could take it? We could take it to the spleen, or we could take it to the liver. And we can use that iron in a whole bunch of different processes. But right now, we only want it for erythropoiesis. So we're going to talk about that right there in just a second. But now, how do we regulate this? I mentioned all these proteins. You're probably wondering, why do I need to know all these proteins? All right, the one I want you to really know is ferroportin. And the reason why is transferrin. Let's come over here to the liver over here. So the transferrin is going to take that iron and it's going to transport it over to tons of tissues. One of them is going to be the liver. And if you look here, there's a little transporter here. This transport protein right here is going to be, or actually a membrane protein, is going to be for transferrin. So when transferrin binds onto this, look, transferrin binds on, and when the transferrin binds on with the iron, it brings it in, right? And then what happens to the iron? The iron can get stored and stuff, right? And we can use it for other processes. But they're in this cell, we have hepatocytes, right? And these hepatocytes, in response to these changing iron levels, it actually can produce a very, very important protein. And this protein is called hepcidin. Why do we need to know about hepcidin? Because hepcidin, what does he do? He controls the activity of ferroportin. So if hepcidin is present, it's actually going to block ferroportin so that it's not so much iron is actually getting into the bloodstream, because too much iron is toxic. So there's one more thing, and this is why I want to mention it. There's a protein here, which is in normal individuals, it's called HFE protein. And guess what this puppy does? He controls hepcidin. So he regulates hepcidin. Well, guess what? In a disease called hemochromatosis, you don't make a functional HFE protein. If you don't make a functional HFE protein, can you make hepcidin? No. And then if hepcidin isn't there, what's going to happen to the ferroportin? Iron's going to start getting loaded into the bloodstream, and then you're going to have iron overload disorder. That's not good. That's why we want to know all of these little transporters. Sometimes the most smallest molecular thing can make a difference to an individual's life, right? So again, that's going to give us that. Now that we got the iron, let's talk about something else, which is going to be B12 and folic acid. There are other things that we definitely need to know. So let's come back over here again. So we got the iron. Let's say we take in lots of leafy vegetables and meats and stuff like that, and that's going to be rich in B12. Or we could take in another molecule, which is going to be called folic acid, which I'm just going to denote with an F. Okay, so we got B12 and we got folic acid. And we take these molecules in. All right, so we take them in through the diet. And they come all the way down here. And they go through the stomach. They go through the duodenum. Okay, we got to stop here at the duodenum now. All right, because what happens is the folic acid, he also gets absorbed across the gut here. So if we have folic acid here, guess where he comes? He can actually get transported into the bloodstream also. So now we got folic acid there. So what do we have here? We have folic acid. So folic acid actually gets transported right across the duodenum, as well as the iron, right? What happens to the B12? This is where we got to be very, very critical and talk about this. In the stomach, there's a cell. And this cell is called the parietal cells. And the parietal cells, they make a protein called intrinsic factor. So intrinsic factor actually binds the B12. So look, it binds onto this B12. It binds onto the B12, and why does it bind onto the B12? Well, let's track this puppy, this little intrinsic factor B12 complex all the way down to the ileum. So if we come down here, the ileum is the last part of the small intestine. So if we come down here, we got the duodenum, then we go through the jejunum, and then we come over here to the ileum. Well, look what's over here at the frickin' ileum. We got this transport protein right here. And this transport protein loves to bind with intrinsic factor. So when intrinsic factor binds here, look, intrinsic factor will change its shape, right? It'll bind on here. So there's our intrinsic factor. And what's the intrinsic factor bound to? B12. So now, look, this B12, it's bound to the intrinsic factor, which is bound to this receptor in the ileum. Boom, it's going to undergo receptor-mediated endocytosis. So it brings it in a vesicle. So here in this vesicle, look what we have now. We have the B12 right here, and it's bound to intrinsic factor, right? Here's our intrinsic factor, this little black protein here. And it's actually a glycoprotein, so it has a little bit of sugar residues on it, right? So again, we have intrinsic factor, which is bound to the B12. Then what happens? It'll actually track its way to the basolateral membrane. And when it tracks its way out, look what's going to happen. The B12 is going to get released into the circulation. And when the B12 gets released into the circulation, there is new hypotheses that B12 doesn't actually travel free circulating. It actually binds here to a protein. And this protein here, this transport protein, is called transcobalamin 1, and it can also bind onto 2. So transcobalamin 1 and 2. And then what's going to happen? That B12, now we have it into the bloodstream. One more thing, and then we're going to go ahead right into this erythropoietic process. I know you guys are excited. Let's come back over here and let's talk about one more thing. Another thing that we need for red blood cell production is we also need certain nutrients. We need amino acids because hemoglobin is one of the main components within red blood cells. And it's made up of heme and globin. Heme is going to be made up of some carbohydrates. So what do we need within our diet? We need carbs. It's also, it looks like I have cabs, sorry guys, carbs. So carbs. So we're going to bring in carbs, and another thing within the heme is fats. We're going to need a lot of fats. And we're also going to need the things to make proteins. What are the building blocks of proteins? Amino acids, right? Or amino acids, right? So again, we're going to have fats, carbs, and amino acids that we're absolutely going to need from the diet. We're not going to go over those mechanisms. We'll do it when we get to the digestive system. But just know that, again, these guys will come into your digestive tract. They'll move down through your stomach. Some of them will actually all of them will get absorbed across the duodenum. And then some of them will have their own transport process, but we're not worried about it right now. So now, what do we have in the bloodstream that we need for this process? We have amino acids. We have carbohydrates. And we have fats. And we have B12 and folic acid and iron. We now have the bare essentials in order for this erythropoietic process to occur. One more thing that we need to talk about before we do this is one really, really important thing. So let's come over here to the kidney. So if we come over here to the kidney, in the kidney you have what's called a nephron that you can see here. This is our nephron. Now, nephron is a whole bunch of kidney tubules that we're going to talk about in future videos, but we're going to take one portion of the kidney, which is the proximal convoluted tubule, and we're going to expand on one of those cells. Now, I told you that the stimulus of erythropoiesis is hypoxia, or anything that causes low oxygen levels within the blood, whether that be due to bleeding, whether that be due to anemia, or whether that be due to you being at higher elevations, whatever. We want to know, how does that affect the kidney now? Okay, so if we look here in the kidney, we have a specific gene here. Okay, you know genes are going to be transcribed and they make mRNA, and then mRNA make proteins, right? Well, there's a very, very important gene there, which is called the EPO gene. The EPO gene. EPO stands for erythropoietin. Super, super important hormone that we're going to have to know here. And I'll explain why. So again, there's this gene here, this EPO gene, or this erythropoietin gene. We're going to refer to it as EPO now because it's easier to write, right, and easier to spell, just EPO. Here's the gene. Well, out here, there's a very important transcription factor, and that transcription factor is called HIF. Which stands for, what does HIF stand for? Hypoxia inducible factor. Right? So that's not bad. Hypoxia inducible factor. So we have this hypoxia inducible factor here. It loves to bind to this gene and synthesize EPO. But we can't have that happening all the time because then we'll make too many red blood cells. So we have to control this. So there's an enzyme here. Look at this guy. Look at this guy here. He's bound onto this hypoxia inducible factor, and he's keeping him fixated so he can't go and bind. So because we don't want him to always bind. But you know that this enzyme, it depends upon oxygen, and the reason why is it actually hydroxylates these enzymes needed. So if we have a low oxygen, will we have hydroxylation reactions? No. And if you don't have hydroxylation reactions, can he hold on to the HIF? No. And if he can't hold on to the HIF, then what happens? HIF gets right into the nucleus, right? Activates and transcribes these genes, and then guess what this puppy starts secreting? EPO, which we already said again is erythropoietin. All right? So far we've gone over all the things that we need in order for this process to occur. Now let's go ahead and show the entire process of this red blood cell formation. So we're going to start here in the bone marrow now. In the bone marrow, we're going to have a stem cell for all the blood cells in our entire body, right? That stem cell is called a hemocytoblast. So we can call it a hemocytoblast, or we can even call it a pluripotent stem cell, or we can call it a hematopoietic stem cell. I'm just going to go ahead and put hemocytoblast. All right? Easy. Now, hemocytoblasts, they're constantly dividing. They're constantly dividing, and when they're dividing into two different cell lineages. One of these cell lineages we don't give a darn about right now. It is important, but we don't care about that one. We're only going to look at this top one up here. So this one right here on the top is the important one that we're going to talk about. It's called the myeloid stem cell. Myeloid. And this one down here is going to be called the lymphoid stem cell. Lymphoid stem cell goes and makes lymphocytes, but we don't care about that right now. We care about the myeloid. So now, this is where I want to get a quick relevance right here. So you know this myeloid stem cell? You know what's really cool about it? This myeloid stem cell can actually divide into 3 different cell lineages. One, it can go into forming platelets. One can actually go into forming red blood cells, which is the one we care about. And one can go into forming white blood cells. So how in the heck does this myeloid stem cell know which cell to turn into? EPO, erythropoietin. So what is erythropoietin going to do? It's going to trigger this myeloid stem cell to start committing itself into making a red blood cell. Not a white blood cell, not a platelet, a red blood cell. So now look, this EPO, where's it going? It's going to come over here, and it's going to drive this step here. Now, this myeloid stem cell, its genes will be activated to start synthesizing hemoglobin, right? So this cell is now genetically pre-programmed for entire red blood cell production. So now, let's go over this now. Look at this. This myeloid stem cell, now we're going to be becoming a specific type of cell. So you know whenever you go into these cell lineages, they always start off with a pro, right? So you always got to start off with a pro. So we're going to put this one is called a proerythroblast. So now I'm not going to write erythroblast for every single one of these, but you know that this is going to be a proerythroblast. What's going to happen is it's going to start, the genes are activated now because EPO has actually driven this step, right? So it's driving all these steps. So EPO is driving these genes to become expressed. What genes are becoming expressed? The genes to make hemoglobin. So now look what starts happening to these genes. The genes start actually undergoing transcription and producing a ton of what's called mRNA. All right, and rRNA and tRNA. It's making a whole bunch of RNA. So now if we look over here, look at this cell now. If we look at this one, it's going to start making a lot of RNA. Well, you know whenever they did these studies, they found that the RNA actually kind of stained more of like a bluer color. It was very, very base-loving, so it's basophilic. So look at this now. So now it's going to have a lot of blue color. And why is that blue? Because there's a lot of ribosome synthesis, and so there's going to be a lot of rRNA synthesis. There's going to be a lot of mRNA synthesis, a lot of tRNA synthesis here. So now this guy is looking like it's very blue. It's very blue-loving or base-loving. So guess what we call it? We call it a basophilic erythroblast. So what is this called? A basophilic erythroblast. Easy, right? Because it loves base. It loves the base. So it's going to turn this blue color. And why is it turning that blue color? Because there's a lot of RNA and a lot of ribosome activity in there. So then, this, you know, with the central dogma, right, of biology, it always goes DNA to RNA. And if it's a specific one, which is mRNA, the mRNA goes to proteins. So now we got to make proteins. So what's going to happen to this mRNA? The mRNA is actually going to get converted into proteins. And what is that protein? Hemoglobin. What color is hemoglobin? It loves this color red. So it looks red. So now look at this. So now here's the nucleus, right? So now what? We're going to have a lot of red here. We're going to have a lot of red. We're going to have some red here. But what's really important to know here is that it's not all going to be red because not all of the ribosomes are completely synthesizing all the proteins. Not all of the mRNA is getting translated. And so if not all the mRNA is getting translated, there's still going to be a little bit of this mRNA left. So now, again, what is this term, DNA to RNA? This is called transcription, right? And then what is this process here called? From RNA to proteins, this is called translation. So there's a lot of transcription occurring in the basophilic part, and there's a little bit of translation occurring here within this part here. It has two colors. So look, it's loving the red and it's loving the blue. So if it's loving two colors, more than one color, we're going to say it loves multiple colors. So it's polychromatic. So it's polychromatic, and it's still an erythroblast. So now, this polychromatic erythroblast, it's going to continue. It's not done yet. It's still got to translate all of this mRNA into proteins. So look at this. It starts doing that, right? Now, you know, in this last step here, this next step we're going to talk about, the nucleus throughout all these processes should start condensing and condensing and condensing. We need it to condense so that we can get it out. We want to get rid of this nucleus and organelles, and I'm going to talk about that here in a second. So now, let's assume that almost all of the mRNA is translated. So let's just draw a little bit of blue here, just a little bit. But now it should primarily be enriched with hemoglobin. So it should be primarily just a lot of hemoglobin. So it should be red, a lot of red here. Okay, so we got a lot of red in here because of the hemoglobin. We might have a little bit of blue here because there still might be a little bit of mRNA left. Now this one is primarily of a specific color, and that is of the red color, right? It's eosinophilic. It's going to be acid-loving. So now this is going to be called a orthochromatic erythroblast. So again, this is called an orthochromatic erythroblast. And again, what is important about this one? The nucleus should be really, really condensing at this point in time. And then a lot of that should be having heavy, heavy hemoglobin by this time. Very, very little mRNA. But then, I told you that the nucleus has to condense. We got to get rid of this nucleus. We don't want the nucleus. Because you know red blood cells are not truly living cells because they lack a nucleus. So then what happens? They eject out their nuclear composition. And they might inject out some of their organelles. What organelles? You know, it has no mitochondria, right? So now look, it got rid of the mitochondria and then it gets rid of this nucleus. Well, what happens? You know macrophages, they love to break down anything that appears to be foreign. So guess what? This macrophage here, he comes in and destroys these contents, these organellular contents, right? Now, it ejects that nucleus out. It's primarily consisting of a lot of hemoglobin. Now what happens? It turns into a specific type of cell that actually gets pushed out into the sinusoidal capillaries. Oh, really important here. There's 3 types of capillaries: sinusoidal capillaries, fenestrated capillaries, and continuous capillaries. The capillaries that we have to know here for the bone marrow is that they're sinusoidal capillaries. All right, so now that we got that, again, this guy, he gets pushed out. He's still going to be in the bone marrow because he's immature, but look at him. He loves it, right? So look at this guy. This is called a reticulocyte. Okay, reticulocyte. So we got this reticulocyte right here. So again, orthochromatic erythroblast pops out its nucleus, pops out its mitochondria, and the macrophage breaks it down. It gets rid of that nucleus so it can primarily hold on to hemoglobin. Then it turns into a reticulocyte. And then that reticulocyte will actually mature eventually into this perfect red blood cell right here. And this is now a mature erythrocyte. So it's a mature erythrocyte. Now, one more thing before we actually finish up this part and go into the degradation pathway. Erythrocytes, they have no mitochondria. And what is the purpose of mitochondria? You know mitochondria love to take the pyruvate, break it down by utilizing oxygen to make ATP, right? Well, it doesn't have that. So can it use oxygen? No. That's what makes this erythrocyte so darn good at its function, is because it doesn't have a mitochondria. And so if it doesn't have a mitochondria, it doesn't utilize the oxygen that it transports. What's the primary function of this erythrocyte? To transport respiratory gases. Like what? It transports oxygen. And where does it transport the oxygen from? It transports the oxygen from the lungs to the tissues. And then what else is it transporting? It's transporting CO2. And where is it transporting the CO2? It's transporting it from the tissues to the lungs. What's another thing? Erythrocytes, they don't live for very long, right? They only have a certain lifespan of about 100 to 120 days. All right, so now, look, we got to figure out also with this stuff, we got to show how does this iron, how does these amino acids, and how do these carbohydrates, and how do these fats, and how does B12 and folic acid, how does these guys all get funneled into this erythropoietic process? Well, if you look here, we could follow this iron, we could follow the amino acids, the carbohydrates, the fats, the B12, the B9, and what is the importance of all these? Let's follow it with just one solid line over here. Let's say that they come over here, and where are they needed? They're needed in these steps here. Why? B12 and folic acid, or B9, are really important for the DNA maturation. So if you don't have B12 and you don't have the folic acid, can the DNA mature properly and condense? No. And if it doesn't mature properly and condense, the actual red blood cells will be super huge. And it's called a macrocytic anemia or a megaloblastic anemia. All right? So that's really important. Now the iron, where does the iron come into play? Well, the iron is going to be needed for this hemoglobin synthesis. You need iron in the heme, right? Or it's not actually considered to be heme. Actually, heme is made up of protoporphyrin, 9, and iron. So if we don't have the iron, do we even really make functional heme? Or do we make functional hemoglobin? No. So if we don't have the iron in these steps, what's going to happen to the actual red blood cell? It's going to get smaller. And if it gets really, really small, it's called microcytic anemia. And again, what do we need those amino acids and those carbohydrates and those fats for? We need the amino acids for the synthesis of hemoglobin. And we're also going to need the carbs and the fats for the synthesis of the heme. So it's so simple, right? Knowing how this all works, how the iron is needed for hemoglobin synthesis. B12 and folic acid is needed for DNA maturation and condensation, right? And then the fats and the carbohydrates and the amino acids are all needed in these steps also. Amino acids are for hemoglobin synthesis, and the fats and the carbohydrates are for the heme synthesis. So it's so simple how now we know how all of this stuff works and fits together. All right, guys, so we finished up the erythropoietic process, and then we're going to get ready to go into the lifespan and the destructive pathway of the erythrocyte. So click up here in the right corner and watch part 2.